Okay, well, so last class we have seen about pollen pistil interaction and we saw about a double fertilization also. Is there any doubt in the sequence of events occurring in the pollen pistil interaction? Any doubts? Okay, the first event is recognition of compatible and non-compatible pollen grains. Once it is a compatible pollen, pollen germination will be taken place. After pollen germination, siphonogamy will take place. After siphonogamy, porogamy will take place. After porogamy, double fertilization. Where double fertilization is taking place. Option A in the ovary, option B in the ovule, option C in the embryo sac. Which will be the right answer?
Embryo sacs.
Option C, embryo
Embryo.
sac. In the embryo sac only it will take place. And the pollen tube will enter at the micropylar end where we have the egg apparatus, which includes the synergids and one egg cell. Okay, now we saw about the compatible pollens which will be accepted. But there are some cases, children, which I want to highlight now. It is called artificial hybridization. To understand, I'll compare this with human being. Insemination is a process where the male gametes are deposited into the female reproductive tract. Artificial insemination is an important event in testy baby program where sperm and ovum are fertilized outside the body in a small culture vial. So hybridization is nothing but crossing between two different plants naturally. But what is artificial hybridization then? In artificial hybridization, we are adopting some scientific technique by which we are enhancing the characters in a newly formed progeny. Therefore, it is a crop improvement program. Grade 10, you have learned about artificial hybridization in CBSE. In grade 12, strategies for enhancement in food resources, you'll be learning about it. It is a crop improvement program in which desired pollen grains are used for pollination. So there are two important techniques that they are adopting that is called emasculation and bagging. Now here you can see here he is doing some emasculation works. Here some artificial pollination is done here. Here some bagging is done. So what about all these things? Number one, emasculation. Imagine a flower is a bisexual. So, what are the different types of pollination they can undergo? If it is a chasmogamous flower, the flower is a bisexual flower. What are the different types of pollination they can undergo if it is a chasmogamous bisexual flower? They will go for autogamy, they'll go for geitonogamy. There's chances of xenogamy also if there is a agents of transfer. But I want only the cross pollination should occur. So what I'm going to do is emasculation is always done in a bisexual flower. It's a one mark question in your NEET also. In unisexual flower, we have a male flower and female flower. There is no need for emasculation. And you all could have learned about genetics. Who is the father of genetics? Yes, come
Gregor
on.
Mendel, sir.
Yeah, Gregor Johann Mendel. The first step did was he actually took a garden pea plant. The flower condition when you take it was actually a bisexual flower. He converted the bisexual flower into a unisexual flower. How means he followed the emasculation technique. So, what is emasculation? It is a technique where the anthers from the bisexual flower bud of a female parent before the anther get dehisces. Therefore, if I take a bisexual flower, if I emasculate, that means if I remove the anther, what will be the condition of the flower now after emasculation? It will be unisexual female flora or unisexual male flora. Yes.
Female flower, sir.
Yes, it will become a unisexual female or pistillate flower. So removal of anther from the bisexual flower bud is called as emasculation. As a result of emasculation, we are getting a unisexual female flower. So bagging. You all have a bag by which you used to come to the school. In the bag, you will store something. And what is inside your bag, it is not known. So when you come to school, there may be some unauthorized material which may be there in your bag. So bag checking will be done frequently in our campus. Bagging means covering up. Here, the flower is now a unisexual or emasculated flowers are now unisexual female flower. There are some chances that unwanted pollen grains also can land because I want to add the desired pollen grains. So here, the emasculated flowers
So
are.
your voice is breaking,
My
sir.
voice is breaking. Just a minute, children.
So,
Yeah. So when you take this into consideration here, so to prevent from unwanted pollination, because pollination is necessary, but to prevent from unwanted pollination or contamination by unwanted pollen grains, we are covering with a butter paper that is called bagging, and we'll put a tag also for identification. So when the stigma attains receptivity, mature pollen grain collected from the anther of a male parent and dusted on the stigma. The flowers are repacked and allowed to develop into the respective post-pollination or fertilization events. So emasculation, bagging, tagging is there but is not there in your book in in-depth manner. So emasculation is done in bisexual flower, where a bisexual flower is converted into a unisexual female flower. In order to avoid contamination by unwanted pollen grains, we are covering with a butter paper called bagging, and we can put a tag for identification. When the stigma becomes maturation or when the stigma becomes receptivity, when receptivity is there in the stigma, then the butter paper will be removed. And in this, you can see artificial pollination will take place. Again, they will rebag it and cover it up. This is called artificial hybridization. There is no need for emasculation in unisexual female flower. So, in unisexual female flower, the first step called emasculation is not at all there. Only bagging is alone is there. The female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using a desired pollen and the flower is again repacked. Any doubt in artificial hybridization, children? Any doubts?
no, sir.
Okay. In-depth knowledge of artificial hybridization is therein. Strategies for enhancement of food resources. In that chapter, we can discuss about that. Now, pre-fertilization events is over for us. We have seen about sporogenesis, micro and megasporogenesis, and we saw about gamete transfer. We saw about the fertilization events, what occurs in double fertilization, all other parts and all we have seen. Now, the fertilization is successful. That means after fertilization, what is the other event? You know that it's egg cell and the central cell with two polar nuclei is there. Don't draw this diagram for your examination, for your understanding. The first male gamete will fuse with an egg cell. Second male gamete will fuse with a central cell with two polar nuclei. As a result of syngamy, we get an zygote. As a result of triple fusion, and primary endosperm nucleus is formed. The zygote will transform to an embryo only when required amount of endosperm is formed because endosperm will nourish the zygote. Afterwards, the zygote will transform into various stages. So, what are the four important events that occurs in post-fertilization events? Means these are endosperm development must takes place. Endosperm is the product of option A syngamy, option B triple fusion. Yes.
Triple fusion.
Yes, it is a product of triple fusion. So the endosperm is required. So only a certain amount of in case of angiosperm, only when certain amount of endosperm is formed, then only we get an embryo. Means then only zygote will transform to a embryo, and the ovule will transform into a seed, and ovary will transform into a fruit. So these are the various events that we're going to discuss. Fine. The first one, you all came to know what is an endosperm. It is a nourishment. So, totally in this chapter, how many types of nourishment tissue we have seen till now? Tapetum will nourish the option
Microscope
A.
mother
Yeah,
cell.
fine. Microspore mother cell. Nucellus will nourish the embryo sac. Option C. Next is endosperm. They will nourish the developing zygote. Next, we have some concept called perisperm, which is remnants of nucellus. About that, we'll be discussing afterwards. So, in the post-fertilization events, synergids will degenerate. Zygote is there now. Primary endosperm nucleus with primary endosperm cell is there. So, degeneration of antipodal cells. Now, we're going to take only zygote and primary endosperm nucleus on the cell. Now, for this to understand, we have to take the example of coconut. So when you take tender coconut, there are two different things. I'll use Tamil now. We'll ask by two different names. Ana தண்ணி அதிகமா வேணும் தேங்காய் அதிகமா வேணும். We used to ask like this. What is that liquid and what is that white kernel means? That liquid is nothing but free nuclear endosperm, and the white kernel of a coconut is called as cellular endosperm. I'll ask a question whether you understood or not. To make chutney, your mom will add free nuclear endosperma, cellular endosperma.
Cellular
That
endosperm.
means you have understood what is a cellular endosperm. So we'll now tell you how the endosperm is formed. The primary endosperm cell, which is transformed from primary endosperm nucleus, they undergo repeated division, simple mitosis only. They form triploid endosperm tissue. The endosperm cells are filled with reserve food material. They used for the nourishment of the or nourishing the developing embryo. Now the next concept, in common, endosperm development, that is primary endosperm nucleus, undergoes successive nuclear division to form free nuclear endosperm. That means in free nuclear endosperm, karyokinesis alone is taking place. Cytokinesis is not taking place in free nuclear endosperm. This can be compared with a three mitotic division in monosporic development of a 1 nucleate, 2 nucleate, 4 nucleate, 8 nucleate. There are also no cell wall formation. Here also there is no cell wall formation initially. If the cell wall formation have taken place, they will develop into a cellular endosperm. So this is called free nuclear endosperm, where only nuclear division undergoing, cell wall formation is not taking place. Number of free nuclei varies greatly. Endosperm becomes cellular. The same free nuclear can transform into cellular only if cell wall formation takes place. For example, the water which we are drinking is nothing but the tender coconut water is a free nuclear endosperm which is made up of thousands of nuclei without cell wall formation. And the white kernel, the surrounding one, is nothing but the cellular endosperm. For the development of zygote, the endosperm may be completely consumed. They become X-albuminous. If endosperm is not completely consumed, they become albuminous. About that, the next class will be discussing about it. Any doubt in endosperm development, children? Any doubt?
Yes.
No doubts? Okay, fine. Now, the endosperm is nourishing the embryo. So, what will occur to the embryo? The zygote have transformed into embryo. The embryo will transform, that is, zygote will transform into a pro-embryo. Pro-embryo transform into a globular embryo. Globular embryo transforms into a heart-shaped embryo. Heart-shaped embryo transforms into a mature embryo, and it is for dicotyledons because they have two cotyledons. It is shown here. You know what is radicle and plumule? If not, again separately I'll be discussing about the structure. So where embryo development is taking place, it is taking at the chalazal enda or at the micropylar enda means it will take at the micropylar end. Why? Because the egg cell is present in the micropylar end. Syngamy takes place at the micropylar end. So zygote is formed at the micropylar end. So embryo development at micropylar end of the embryo sac where the zygote is situated. More zygote divide only after a certain amount of endosperm is born because it gives nourishment to the developing embryo. In monocot and dicot, seeds differ greatly, but embryogeny, that is early embryonic development, is similar. Now we're going to see that the embryo undergo into pro-embryo, globular, heart-shaped, and mature embryo. Now next we have to still look about the structure of an embryo. So this is the structure of a monocot in DNA. This is the structure of dicot in DNA. I require my stylus pen to go ahead with the concept. Now I'm drawing this as an imaginary axis. I'm drawing this line as an imaginary axis. So what does it have? That is the first point we're going to discuss. It has an embryonical axis. I'll just make it a little bit bigger. It has an embryonical axis and two cotyledons. What is embryonical axis? The imaginary line which I have given you here. It is an embryonical axis. How we have to identify the embryonical axis means based on the level of cotyledons only we're going to discuss. So the portion above the embryonical axis is called as the epiquartile, which is not represented in the diagram. So this is the epiquartile region, and the epiquartile region terminates at plumule. The plumule will transform into the shoot tip. The portion below the embryonical axis is the hypocotyl, ends at radicle, and radicle is nothing but developed into the root system. It is covered by the root cap. So, in a dicot embryo, what is an important event? In a dicot embryo, the very important thing is we have an imaginary axis called as the embryonical axis, which differentiate means the portions above the embryonical axis, that is above the level of cotyledons, is called as the epicotyle, terminates at plumule, develop into the shoot system, that is the stem tip, and the portion below the embryonical axis is called as the hypocotyle, which terminates at the radicle, which develop into the root system covered by a root cap. The next is monocot. So dicots, they had two embryo children, but monocots, what about them? They have only one cotyledon in case of monocot. In grass family, the monocots cotyledons is called as the scutellum. Scutellum is nothing but the cotyledons. Now here we have the imaginary axis called as the embryonical axis. So I'm going to draw the embryonical axis here. So this is the embryonical axis, the imaginary line. So, excuse me, I'll take the, yeah, fine. It is okay. So we know that scutellum is nothing but there is only one cotyledon. It is called scutellum in case of grass family, and grass is a monocot. It is situated lateral to the embryonical axis. The lower end of the embryonical axis is called as the radicle, and radicle is covered by the root cap, and it is covered by coleoriza, which is an undifferentiated sheath. What is the difference between sclerosa? About that we're going to discuss separately. Next is the portion of the embryonical axis that is above the level of cotyledons, above the level of attachment. We have the epicotyle, which terminates epicotyle and have the shoot apex, which have the shoot apex, and if some leaf primordia can be seen here and here and there. Epiplast we have learned about in grade 11, and it is enclosed in the coleoptile and hollow foliar structure. So, what is the main difference between a coleoptile, which is given here, and a coleorhiza, which is given here? Some people say coleorhiza also. So, look about your coleoptile and coleorhiza. What is a coleoptile? In canary grass experiment in discovering of auxin by Charles Darwin and his friend son Francis Darwin, we have discussed about how the auxin was discovered. It is a sheath protecting a young shoot tip in grass or cereals where we have enough amount of auxin. It is a sheath protecting the root of a germinating grass or cereal grains, and we have, it is actually a protective sheath, and it is an undifferentiated sheath. It covers the plumule, which develops into the shoot tip. It covers the radicle and the root cap, which develop into the root tip. It breaks the seed coat and it elongates the generally coleoptile. And when you see, it comes out from the soil and turns into green. But when you look about coleoriza or coleoriza, it breaks the seed coat, but but stops further growth. It elongates the growth, stops the further growth, and it will remain inside the soil.